Okay guys, welcome to the lesson on Archaea. I've given you this diagram on the LMS page so you can print it, or if your printer's not working or if you want to save ink, then you can just simply examine it. So I thought I'd just take a couple minutes and go over this with you because now we are truly starting diversity in the way that we're going to be covering the material related to different domains. Okay, so what do I mean by that? We've got all living things here. Okay, so every single thing on Earth is represented in this box. All right, and then so from there, you've learned that domains were added after many, many years of having classification with Carolus Linnaeus. And domains were added because scientists realized that bacteria and archaea were not the same thing. So originally we had archaebacteria, and they were classified as bacteria. And then when DNA evidence became available, we realized then that archaea and bacteria have quite different DNA. And honestly, we used to teach that eukaryotes came from bacteria, but we came from archaea. So this DNA evidence that's come into light has really changed things up. And so they added this domain above the kingdoms. All right, so what do you need to know about that? All living things are divided into 3 domains. Okay, we've got bacteria, archaea, and eukarya. Now, today and tomorrow, I'm going to be exploring bacteria and archaea. So today I'm starting with archaea. So archaea are here, and notice this, their domain name is the same as their kingdom name. Okay, so I'm just going to highlight, normally I would highlight the whole box, but that's good. Okay, so we've got the domain archaea and the domain bacteria. Now I'm going to highlight these guys in the same color, even though obviously they have different names. So the domain bacteria starts with the kingdom bacteria also. So these guys only have one domain, one kingdom, one domain, one kingdom. Okay. Why am I highlighting them in the same color? Because these guys are your prokaryotes. Okay, so just excuse the slanted writing. I've actually got like a phone in front of me right now so I can videotape you guys, or videotape this lesson. So these guys are prokaryotes. Bacteria and archaea are your prokaryotes. And on this side, we have a different domain called eukarya. And eukarya has 4 kingdoms. Okay, so not just one. It's not a 1-to-1 ratio like it is over here with these prokaryotes. And eukarya contains eukaryotes. Okay, and what's the difference? These guys very simply have their DNA surrounded by a nuclear membrane. Okay, so we call that a nucleus, but really at this point you should know that means that their DNA is surrounded by a membrane, okay, a plasma membrane. These guys, the prokaryotes, don't have a membrane surrounding their genetic material. So their genetic material is just free floating in the cytoplasm or the cytosol, okay? So what do I want you to know from this diagram? All living things are broken into 3 domains, okay? 2 of those domains are prokaryotic. 1 of those domains is eukaryotic. The prokaryotic domains only have 1 kingdom each, named the same as the domain. Okay, and on here, you're going to see many of the characteristics as we go through. Okay, as we go through, you're going to see many of the characteristics I'm covering in the notes. Okay, so this is a really good summary page for you. I would have it in front of me whenever I'm being tested. Okay. All right, and just over here, so this is going to be today's lesson, which is Wednesday. This is tomorrow's lesson, which is Thursday, and then you have your quiz just based on these 2. All right, I just had to come up with a way to divide the quizzes. I said to you guys you're going to have 2 to 3 quizzes per unit. We're not having any unit tests, okay? So I figured just quizzing you on the prokaryotes would be a good way to break this up. And then you'll have a quiz on the eukaryotes, okay? And then in addition to that, any questions I've given you on diversity and classification, the first couple of textbook readings, those will also be on your quiz, okay? So, so this is Wednesday, Thursday, quiz Friday, and then on Monday we will start with Protista, okay? And then we'll go into these guys. All right, so that's a very good thing to have printed out. And so today I'm going to go through Archaea with you. So I've tried to make this very simple. The note is quite short, which is good. it just goes through the most important characteristics. So in other words, everything on here is important. So here we go with the domain Archaea. The domain name is Archaea. The kingdom name is Archaea. What's important here is that you've got prokaryotes. Prokaryotes do not have genetic material bound in a membrane. Okay, so the genetic material, which you can call DNA, genetic material is not membrane bound. Okay, so in other words, there's no plasma membrane, there's no nuclear membrane surrounding it. All right, what's also important to note here, these are distinguishing characteristics between Archaea bacteria and the eukaryotes, is that Archaea are unicellular. So in other words, when we get to the eukaryotes, to kingdom or to domain eukarya, many of those organisms are going to be multicellular. Unicellular means these guys are one cell large. Okay, so one cell large. How do they get their nutrition? So in other words, how do they get their energy? They can be autotrophic or heterotrophic. So just give me a moment here. I'd rather write in pen. Okay, so we've got autotrophs. These guys can make their own food. Or I'm going to say make their own energy. So they're going to convert one energy source to another energy source. Capital E stands for energy as we go forward, okay? Heterotrophs, and I'm sure that you know this even from elementary school, but it's a good thing to be reminded, right? Heterotrophs have to consume. And so when you say, okay, I'm going to go make my dinner or my breakfast, you actually have to consume something. So that's why you're not classified as an autotroph. Yeah, you can make your own meal, but you have to eat something else, okay? These guys can just convert one form of energy to another one. Kind of what we're doing, but we're consuming along the way. So that's the distinguishing point between those two. And then means of reproduction is asexual, okay? So there's no mating, in other words. One organism splits into two. So one organism, and in this case, because they are unicellular, we're going to have one cell, one organism splits into two identical organisms. Okay, so times two. So one organism splits into two identical organisms. All right, and so when I quiz you, these are going to be many of the things I'm going to be looking for in the multiple choice, okay? Because this is what distinguishes Archaea from bacteria from eukarya, all right? So right here, these are very important points. All right, so now we're going to just go through what's the difference between Archaea and bacteria. Well, first of all, their DNA is different, okay? I didn't list that there, but they absolutely have different DNA, as do you and I. But the differences between these guys put them into different domains altogether. Okay, so rather large differences. Archaea live in extreme environments that bacteria cannot inhabit. So you don't need to be worried about these guys. They're not interested in us because they are living in environments that other organisms can't even imagine living in. Okay, so things that bacteria can't take advantage of, Archaea can take advantage of. All right, so they live in, I said, in extreme environments that bacteria typically do not inhabit. This is going to become important today. It's not going to make any sense to you. But Archaea do not have peptidoglycan. So peptidoglycan is a protein sugar polypeptide. It's a very long molecule, and you're going to find it in cell walls of bacteria. All right? And as I get into bacteria tomorrow, I'm going to tell you why that's so important. But you do need to know Archaea don't have the gene to make peptidoglycan. So they're not making this compound in their cell walls. And again, that's going to become more important tomorrow. It's believed that eukaryotes branched off from Archaea approximately 1.7 billion years ago. So I thought I'd put this in there because what I want you to know is that our mRNA sequences are more similar to theirs than to bacteria. So that's the difference between archaea and bacteria. At this point in 2020, we believe that we evolved from archaea and not from bacteria. What are mRNA sequences? These things are, it's RNA, okay, just a quick little explanation of this. I could say DNA, and if you want to write DNA in there, then you'd understand it better. But DNA is too big to leave the nucleus. And so when you're making protein, you have to make this copy of your DNA called an mRNA, a messenger RNA molecule. And mRNA is single-stranded. So mRNA is allowed to leave the nucleus. And so when it leaves the nucleus, then it gives the instructions on how to make proteins, okay? So basically mRNA is a copy of DNA. mRNA can leave the nucleus, DNA cannot. So if that's too much, just think, okay, my genetic material is much more similar to archaea than it is to bacteria. And that's not to say that we're not similar to bacteria, it's just that we're more similar to archaea. Okay? And finally, archaea cannot be killed by antibiotics because antibiotics are designed to kill bacteria. Okay? And like I said, don't worry about archaea. They're not infectious to us. It's not something you need to worry about. They're not trying to compete with us like bacteria are. Bacteria love us. They love our food. Archaea aren't interested in us because they've opened up a whole bunch of habitats that nothing else can live in. And so they've got all these environments where they can thrive. Okay? So as we progress through this unit, I'm going to give you ways that all these different kingdoms, phyla are organized. So classification, taxonomy, right? So I'm going to give you ways in which we classify the different organisms in each domain, kingdom, etc. So archaea is divided based on where they live. So this is a very easy one to understand. So scientists have divided archaea based on where they live, so based on their habitat. So the first type of archaea that we're going to look at is a methanogen. Okay? Why is it called a methanogen? Because it produces methane. Okay, so we can highlight that. It produces what some people will call marsh gas. Marsh gas is methane, or intestinal gas, which is methane. So in 2D, we talked to you guys about climate change and cows producing methane gas in their intestines. This is what's responsible for them. Okay, so it's not bacteria so much. They have archaea in their intestines because archaea live in an anaerobic environment, and the intestine of a cow is anaerobic. Okay, so anaerobic means having no oxygen. If you have the word aerobic, it means it's an oxygen-rich environment. If you put an AN in front of it, it means it's not oxygen-rich, so no oxygen. All right? Methanogens obtain energy by changing hydrogen and carbon dioxide into methane gas. So you and I can't do that, and that's why we are not classified as autotrophs. These guys change hydrogen and carbon dioxide into methane gas, and that's their waste product, okay? When they're doing that, this is the waste product, but they're getting energy from that conversion. Where are you gonna find them? Swamps, marshes, sewage treatment plants. So when you flush the toilet, what's in your toilet goes down to the sewage treatment plant, and people actually culture methanogens to help to start the decomposition process and the digestive tracts of cows, okay? And so why are they in the cow? Because they're helping to break down cellulose for herbivores. So you and I can't digest cellulose. Okay, we'll talk about that when we get to the next unit. We used to be able to with our appendix, but the appendix doesn't serve that purpose anymore. But cows are still able to chew grass and digest cellulose. All right, the next group is the extreme halophiles. So when I see halophile, my brain automatically associates it with salt, okay? Why do I do that? I think it's because I see this AL in halophile and I see the AL in salt. So if that helps you, use it, okay? So extreme halophiles live in very salty water. So we're talking about like water that bacteria can't live in. So you have to think about this, okay? Just a little backstory. When Europeans came over on ships and boats, they brought with them food to sustain them on that long journey because traveling across the Atlantic Ocean was not a short journey as it would be now, okay? So what they did with the food that they had was they salted it. So if they had meat, that's why when you have cold cuts, they're very, very salty. Because if they had meat, they added lots and lots of salt. They did that so bacteria couldn't survive on them. Because as soon as bacteria gets a hold of our food, bacteria starts to decompose it for its own purpose. It's getting energy from it. And we can't eat it anymore because the bacteria count gets so large that we would get sick. and the bacteria would start to grow inside of us, right? So that's why I'm trying to tell you here that bacteria cannot compete with archaea in the same environment. The environments that archaea do well in, bacteria do not do well in. So one of those environments is very, very salty water. Okay, so for example, if you went into the Dead Sea, you would find no bacteria because they can't handle that salt, but the halophiles can certainly handle it, and they're of no problem to us. Okay, so we don't have to worry about them being there. Another example is Great Salt Lake. So any place that's very, very salty. Why? Because they're using salt to generate ATP. Okay, so I just put in brackets here, energy source that cells run on is ATP. So you eat food, you get calories from food, but you don't run on calories. You have to change all of your energy from your food into something called adenosine triphosphate during cellular respiration. Okay, and so that's the energy that we run on. It's like the gas or the fuel for our cells. And this is how they get their nutrition. All right, so I'm just referring to nutrition there because up here I talk about nutrition. So these guys are autotrophs. All right, thermoacidophiles. Thermo means heat. Okay, thermos, right? Heat. And acido should be very clear, acid. So our last group of archaea live in very acidic environments that are extremely hot. So they live in, that should say hot, I just rewrote this note, so that's a spelling error. Live in extremely hot, anything at 110 degrees Celsius and above, water, and acidic water at a pH of 2. Okay, another thing that people do is they can their food, right? And when you can your food or you pickle your food, typically when you pickle it, you're going to add vinegar. Why do you add vinegar? Because you want those cucumbers or whatever else you're pickling to survive the winter in that jar. Bacteria don't like acids. They don't like pH environments of 2. We kill them off in our stomachs at our pH of 2. Vinegar kills off bacteria. They can't survive it, but archaea can certainly survive it, and they thrive in acidic water at a pH of 2. Okay, so where are you going to find that on Earth? Well, one of the best examples is in Yellowstone National Park. Okay, so they've got volcanic vents, all right, and they've got hot springs. So they're found in those hot springs, extremely hot. We'd also find them in volcanic vents on the ocean floor and on land. Okay, so I just gave you an example here. And I said that this leaks scalding acidic water, and nothing else could ever occupy that space, but certainly a thermoacidophile can. Okay, so that wraps up your questions or your note on archaea. What I want you to do are these 5 questions for understanding.